Thank you so much. I mean, it was also great to see Bob here. I was, one of his postdoc students as well. And I think, I don't know, whoever kept me after, a talk from Bob and Jennifer Doudna, I think, no pressures, although I think this is too much pressure. But I'm glad to be here. Thanks again for your invitation. And so this picture that you see right now is basically, we're talking a lot about AI. So I asked AI to actually make a lipid nanoparticle with mRNA, but as a, sort of a artistic rendition. And this is what it came up with. And it is very interesting because, I think there has been a lot of talk about AI. And I think at least, I don't know if it'll make better LNPs for us, but at least it's coming up with like very good pictures. So my lab is located in Portland, and I'll talk today about the quest for the next generation lipid nanoparticles for cell-specific delivery of RNA. These are my conflicts of interest. I am a co-founder of Enterex Bio and also RNAvax Bio and SAB member to a couple of companies as well. So, as Bob mentioned, we have, basically moved from sort of a Renaissance time to an industrial revolution. And as you can see here, it was a lot of like surreality in as these things started to come into the main picture. As you can see here, the New York Times had all these articles that were coming in, how to ship the vaccines. There were, markets that were getting changed based on the news about vaccines. And it led to the culmination of a Nobel Prize for Katie Carico and Drew Weissman for modification of mRNA. And again, since Bob and others have already covered about the use of RNA, I'll not go in much detail, but I think, the main part is that you could basically have a rapid transient effect, and that can be utilized for various applications. One of the key hero in this story has been a lipid nanoparticle. Like Bob mentioned, it contains an ionizable lipid, which at low pH can interact with RNA. Then you can add different lipids together, like helper lipid, cholesterol, and PEG lipids, to basically make a spherical LNP, which once exposed to cells get internalized inside the cells, and then the ionizable lipid again converts into a positive charge from neutral, causes an endosomal escape, and then you can see a reaction, which is basically translation that happens. And now we have started to understand a lot of structure relationship of ionizable lipids. They contain these quaternary or tertiary or primary amines, which give the charge. At low pH, there are different linkers that give biodegradability and different tails that give hydrophobic interaction to form LNP. And so these can package and protect nucleic acids. They have a large cargo capacity, biodegradable. Scale has become a completely different meaning because now you can make billions of this. So if you can make a particle that can go to the brain, I think the clinical stuff for that is already realized. And they're modular in nature, and also they're approved for sRNA delivery, now for vaccines, and there are companies like Intellia and Verve who are doing clinical trials for gene editing in humans as we speak. So LNPs have been, sort of best in class at this point to deliver some of these nucleic acids. But sometimes people think that these things are just, sort of not active ingredients, but just excipients. So this is just to show you an example. These are galectin-8 cells, which have stably transfected GFP in them. If you throw in some lipid nanoparticles containing cholesterol, the galectin-8 starts to go into the endosomal vesicles, and you'll start to see escape events because the endosome has broken up. And if we add like cytosterol instead of cholesterol in a case, you see even massive endosomal escape that has happened. So what I'm trying to suggest here are that we can make hundreds of libraries of these lipids, but there are only few of them that actually work because there are only few that in vivo can cause an endosomal escape, have the release of the RNA that goes to the cytosol and works. So, these are not benign sort of molecules, but they are actively engaging with biological processes. But today I would, focus our, research focus is moving beyond the tissues that we can hit. For example, if you give an LNP IV, it goes to the liver. So there are a lot of trials and that are happening for liver-based diseases. Vaccines, of course, there is the muscle, but we have all these other organs that we can deliver. And if we can, there are already disease targets that we can start moving into. And as I said, if we find the right compositions, that would be transformative for the field. So this is a recent sort of idea that we have proposed in Nature Nano, where I think some things have changed in the field. I mean, usually we used to make LNPs through very cumbersome ways. Now you can make a 96 well or a 384 well with high throughput formulation of these LNPs. Then you can attach targeting ligands, and that also has been done for a long time. But in this fashion, you can have different avidity and different targeting moieties on LNPs. With that, you can add different genes, or DNA-based barcodes in all these particles so that you don't have to test one by one. You can mix all these particles together, inject 30 of them, find which material goes in where, and then do some sort of a directed evolution of nanoparticle formulation. So our hope is that now we'll start using some new things that have helped the field, including barcoding and high throughput formulations, to start directing evolution of nanoparticle formulations in the future. But while we wait for that, there are a couple of things that we have started to focus on. One is we are doing local delivery into the eye, and again, as Bob mentioned in his talk, they were finding things that have already worked for AMDs, but our interest is towards inherited retinal degeneration. So eye is a beautiful organ, composes of a neural retina. So once the light passes through the lens, it reaches the retina, and it's basically, the cross section of a retina upside down. So the light basically enters through the ganglionic cell layer, moves into the photoreceptors where light signal is modified into an electric signal, which then forms an image into the brain. The photoreceptors are lined with an epithelial cell layer. It's also called the liver of the eye because they're very phagocytic. Every day when you wake up, these outer segments are taken up by the RPE cells. So in case of inherited retinal degeneration, what happens is that the photoreceptors start to die. So either there can be a mutation in the RPE or the photoreceptors. And the fundamental difference that happens is that the photoreceptors are basically start to die off. And what happens next is you basically start to lose your peripheral vision. So this is Polo, a graduate student of mine who now is a postdoc at Stanford. Marco, made this image that if photoreceptors start to go off, you stop, visualizing Polo after losing some of the vision from the periphery. So our agenda was that can we deliver lipid nanoparticles to the back of the eye, especially where AAVs, although have been successful for products like Luxturna, which give a gene back to RPE. The challenge is there is cargo size limitations with AAVs. They can't package more than 4 kb. There is immunogenicity with them, but with LNPs, we can carry large cargo, including prime editors, which are large in size. And we might want limited expression of things like Cas9 because we don't want it to last longer, like what happens in the case of AAVs. So there are a couple of ways you can inject into the back of the eye. One is a direct, subretinal injection. So you take and poke a needle and it form a bleb, and you can form multiple blebs in the eye. It is a directly, it's a direct injection, but it's highly invasive. It detaches the retina. It is also dose limiting. The other way is you can do an intravitreal injection, but again, the vitreous is very viscous and there is low tissue penetration. People are also trying suprachoroidal routes of administration to hit back into the eye as well. So when we started working in this around 2014, when I reached out to a new professor, Mark Penesi, who was working in the IRD space. And Mark now actually has led the Editas's clinical trial in the eye and is leading a lot of clinical trials in the eye, along with Renee Reyes, who's now a professor at OHSU. She was a senior scientist then, and with my grad student Sid Patel, who also is in a biotech company in Boston. So we took the lipids that are already well established or were already well established at that time. I think MC3 was not approved for siRNA delivery, but we grouped them based on their different chemistry and ionizability. And what you see here is that all our particles are basically transfecting, and this is GFP expression in the RPE, so the liver of the eye, which is very phagocytic in nature. To show the robustness of our work, we then injected these particles in AI9 mice, which have a LOX-P site next to a TD tomato. And then if you give a cre recombinase, it activates it. So if you look at the fundus image, so direct, like looking into the eye of a live animal, we see this TD tomato light up. If you then take the retina out and then cross-section it, you basically have RPE, lights up again. So what I'm trying to say is that independent of how we did our experiments and in which models, the expression was limited to RPE, intravitreal injections were not going to the back of the eye. So we started working in this area, and another postdoc of mine, Milan Gautam, what he did was he started to modify the surface of these particles, making them positive, negative, and neutral charge by just modifying a little bit of PEG. So there is just 1.5% of PEG in these formulations, and he started to change that. So 1.2% of the conventional PEGs, but 0.3% of either a positively charged, neutral, or negatively charged PEG. So as you can see here, this is a neutral charge PEG, lipid nanoparticle, positive and negatively charged particles. They all form core shell morphology and they are spherical in nature. So not much difference on that case. If you inject these now, and and what you find is that the neutral and positively charged particles are all again located into the RPE, right? But the negatively charged particles, the X and the Z, we start to see this beautiful photoreceptor expression, which again has taken us a long time to get here. We can actually make this a bigger image. And as you can see here, you see RPE, beautiful, outer segments and photoreceptor expression that ensues after that. We then took CRISPR-Cas9 based systems, used a Cas9 RNA with a guide, made these particles, injected them, did the fundus imaging, but also we took the whole retina and did the RPE flat mounting and next generation sequencing to visualize if we can actually get genome editing in an AI9 model. And as you can see, again, we do find that, we have a high intensity in once we gene edit the AI9 model. We also see expression in the RPE flat mount, but we're still figuring out how we can see a better expression in a non-dividing photoreceptor cells because the charge of these particles also changed. Recently, we have published so all these particles were being tested with, known lipids like MC3, and they have some sort of an immune reaction even within the eye. So that's why the field had not moved much in this space. So what we started to do was build our own library and then pan for efficacy in delivery and immunogenicity. So we made these Gewalt-based lipids. They have this thiophene core, which gives us huge flexibility. You can make different tails. You can make a single lipid tail, double lipid tail, triple lipid tail of these particles. You can change the ionizable group on the amine here, but you can move that as well. So you can make a tail on this functional group and then move the ionizable group on completely different ends. So this gives us a lot of rational design that helps. And what we found was if you inject this guy, it goes to the lung. These guys were going into the liver. But more importantly, when we injected again a non-human primate with some of our particles, we see this beautiful photoreceptor transfection, very clean and clear transfection. And then when we take this and use immunosuppressants, because those are used in a lot of trials with AAVs, what we find is that at 225 microgram of RNA, you do find some sort of immune reaction that happens, but with an immune suppressant that basically goes away. More excitingly, what we find is, and this is, we have published it, that there is a higher expression somehow when we are using an immune suppression agent. So at 2.5 and 25 microgram, even at 2.5 microgram, we are seeing very high expression of GFP. And these are around 30 to 40% rods and similar amount of cones that we see the expression in. With Marco, again, what we did was we also wanted to find particles that basically can cross the vitreous barrier. We took a phaged library with different 7 mers of the phages, injected them directly into the eye, identified those phages which were crossing through, identified those peptides, coated the particles on, and then at this point, just subretinally, we have shown photoreceptor expression, and hopefully in the future, we'll try some intravitreal injections with these particles as well. We have also, and this is unpublished data, where we have made the LNPs, and because we thought that the surface modification of LNPs was effective in delivery into the eye, what Yulia, postdoc in my and now a senior scientist in the lab, did, she made a PFV or a preformed vesicle and then coated an mRNA on top of it. Usually we think the only RNA would work if it's encapsulated. But here, what we find is that even if you make a particle that has an mRNA on top of it, you form these blebs. And that can, what we did was we wanted to test them in stem cells. So we took iPS cells and converted them into retinal organoids so that we can test a lot of guides if we are going after some diseases of the eye like USH, and identify which guides work the best. But rather than cell models, we are looking to do that in retinal organoids. So of course, you again see this beautiful PFV expression of the RPE, but also the expression in the photoreceptors. And finally, what we are also trying to do is delivery to different routes through the lung. And in this case, a postdoc of mine, Senya, what she did was she has made she has worked with a collaborator and has tested. So we are not just limiting ourselves to lipids. This is a polymer library with like 150 polymer lipid hybrids. She tested them to see if they can show endosomal escape and figured out which particles actually end up showing endosomal escape. And from that, we started to understand some variability that was happening, and hopefully I can talk more about it. This is still undergoing some patent protection. And through that, we started to understand the structures, and that led her to make particles that could actually, do a very high level of gene editing as compared to other, systems. We see the editing happening in endothelial cells and B and T cells at this point. And, she did a lot of multiple IHCs, molecular biology, and FACS analysis to really get robust data in the lung space as well. What we are also doing is we are basically using a new chip to help in nebulization of some of these particles, because if you use materials that are in a using a vibrating mesh nebulizer, they can break apart. So this, we formed a company called Rare, which works with Funai. So it basically takes ink based cartridges and has developed that to form these plumes. that again can be digitally controlled, again have a very good ability to maintain the size and shape of the particles. And this again work is under revisions at this point. And we find that we can actually hit the lungs pretty nicely with some of these new chip-based materials as well. So finally, I think I'll thank all my lab members, current collaborators, my mentors, Bob Langer, and I see Dan has showed up as well, who have always been very supportive. Also, this is my daughter showing as when she was 7, showing this, poster for the COVID vaccine. And I think we start recruiting people early so that they can join the lab. And this is my lab in Portland. And finally, again, I asked the AI program to make an LNP with an RNA with a picture that I provided, and then to make a modern art out of it. So, like I said, I don't know what the role of AI would be in LNP development, but I know one thing for sure: it makes very nice pictures. With that, I think I have some time for questions.